Lucifer comes from the Latin words luce, meaning light, and fere, meaning to bring or to bear. He was considered to be the planet Venus, the morning star. Luciferin is named due to its unique ability, like the first star of the morning, to bring light out of darkness. The oldest written reference of bioluminescence, the creation of light by living organisms, was by Aristotle. 2,500 years ago, he was already ahead of modern scientists by simply noting that these living creatures could produce light without creating any heat. The Roman scholar Pliny the Elder examined a clam that could still bioluminesce even after its death, much like Pliny's work remained long after he was suffocated and then crushed by Mount Vesuvius's spastic regurgitation. Records of bioluminescence are scant after this, only once more reappearing 1,300 years later in medieval Italy. In a period of oppressive theocratic rule and copious amounts of unwashed masses, fireflies were believed to be the souls of the dead roaming the earth. This caused much paranoid distrust of very small glowing things for many centuries. The Age of Enlightenment finally reinstated scientific studies and an interest in glowing organisms. The advent of the microscope in 1625 allowed certain scientists to discover that glowing, decaying animal bodies was due to bioluminescent bacteria feeding on the corpses and not the devil. In 1667, scientist Robert Boyle proved that removing air from a bell jar causes fungi to stop glowing. Sort of like when you stick a person underwater, he stops breathing. But Darwin summed up this progress in his Origin of Species when he wrote, "It is impossible to conceive how these wondrous organs have been produced." Bioluminescence did not fit his theory of evolution, so he tossed it into the category special difficulties. It wasn't until 1887 when a scientist named Raphael Dubois discovered the chemicals that caused bioluminescence. He named the catalyst luciferase and the fuel that produced light luciferin. Other scientists spent another 70 years isolating and purifying the chemicals. By then, luciferin and its potential uses had moved into mainstream popularity. When Aristotle noticed that these living lights did not produce heat, he had hit on perhaps the most fundamental aspect of luciferin, its structural efficiency. Because light is created solely through a mixing of chemicals, no heat is involved. This chemiluminescence is considered a cold light. By developing chemical light, organisms are able to use this ability without a huge tax on their systems or energy consumption. In some creatures that continually glow, cold light proves to be a distinct advantage. Luciferin, in its pure state, emits a green, yellow, or blue glow, but many creatures are capable of different colors, sometimes up to 3 or 4 within the same animal. This is only possible with fluorescence. Fluorescence is the conversion of one color of light to another. It relies on photonic wavelengths to absorb one color and emit a different one. Luciferin can therefore be found in every color of the rainbow. There are at least 5 different molecules that fall under the term luciferin: dinoflagellate, vargulin, colenterrazine, bacterial, and firefly. But since each class of genera developed their own unique methods, there are possibly more varieties yet to be found. Dinoflagellates are a type of plankton found in salt water. The molecular structure of dinoflagellate luciferin is similar to and is believed to be derived from chlorophyll. Dinoflagellates bioluminesce when the surrounding pH falls to 6 or when physically disturbed. Another set of marine creatures, the shrimp vargula, or sea fireflies, have another molecular form of luciferin. A variety of other marine creatures use colenterrazine to glow. These include many squid, shrimp, some fish, and plankton. Bacterial luciferin allow different bacterial species to either glow in response to stimuli or maintain a continuous luminescence. Continuously luminescent varieties often share symbiotic relationships with fish and other animals. Firefly luciferin may be one of the most direct experiences many of us have with bioluminescence. These insects have the simplest system by manufacturing their own luciferin and mixing it with only oxygen and the catalyzing enzyme luciferase. The bugs are therefore the best representation of how light is created. The 3 essential ingredients of bioluminescence are luciferin, luciferase, and oxygen. Luciferase is the energy storage unit. Without this enzyme, luciferin would have no fuel to produce light and would sit alone and useless in a corner of the cell. But most catalysts require 2+ ions in order to start a reaction, meaning the fuel requires fuel. Therefore, magnesium or calcium ions bind to the protein. Luciferase begins madly breaking ATP into AMP. As heat is the source of energy in incandescence, AMP acts as the process's chemical energy. Luciferin then binds itself to AMP, which is the reduced form of ATP. Next, oxygen molecules bind to the substance. AMP then attaches to the hydrogen, freeing it from the luciferin and releasing energy. The end product is carbon dioxide, oxyluciferin, and light. This manner is so successful because all varieties of luciferin are very unstable. The term luciferin covers all chemical interactions that result in bioluminescence. This means that there are many different ways organisms have evolved to create light. Scientists originally believed that all bioluminescent mechanisms developed from one common ancestor and spread through the animal kingdoms via symbiotic relationships with luminous bacteria. This was disproven when researchers failed to produce light from mixing luciferin of one species with luciferase from another. On top of that, bioluminescence pops up sporadically throughout time and geographic space, with little to no connection between the species. Out of the 17 phyla, 11 of them have some random organism that is capable of glowing. Scientists therefore surmise that bioluminescence evolved separately and repeatedly. A glowworm in the caves of New Zealand figured out one way to produce light, while a squid in the Black Ocean depths developed something completely different. They both produce blue light, but the way they learn to go about it is like breathing air versus breathing water. Luminosity is, not surprisingly, found almost exclusively in animals that dwell in perpetual darkness. About 90% of the creatures that exist deep in the ocean have some form of luminescence. Others, such as fireflies, are only active at night. Bioluminescent organisms have also found two methods to luminesce. Either they create it themselves, or they share a symbiotic relationship with luminous bacteria. Those that create light through their own processes are able to manifest it in two different ways: intracellular luminescence and extracellular luminescence. The intracellular method produces light within the cells of the body, which then escapes through its skin. In the extracellular method, organisms often separate luciferin and luciferase into two different glands, and then squirt them both out into the water at the same time, allowing the chemicals to mix freely while the organism often escapes. Since scientists have been able to synthesize luciferin and luciferase, they have found many ways to implement these chemicals for medical and technological use. Bioluminescence has greatly contributed to understanding and treating diseases. But more importantly, it has also revealed how the human body works in more detail than ever before. The microscope revealed cells. Bioluminescence reveals what is going on inside them. In genetic research, the DNA that codes for luciferase is attached immediately following a promoter codon in a protein. The promoter then tells the RNA in the cell to create a transgenic protein that glows if exposed to luciferin. Researchers are able to target very specific proteins and thus can track their movement, development, and interactions. This is called tagging. Scientists tag proteins for multiple reasons. One area of study is the interaction between organelles in the cell. Bioluminescence has captured the curiosity and fascination of human beings for thousands of years. A little over 100 years ago, luciferin's molecular secret was cracked, and the resulting research has had a growing and dramatic impact on the way we understand how the body works. The fact this molecular process requires so few components, yields high efficiency, and evolved repeatedly across the globe simply adds to the amazement that nature has figured out a way to create light billions of years before man. And she did it better.
Go, little glowworm, fly up, fire, glow like an incandescent wire. Glow